I'm Allison Johnjack, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual Brown Bag Lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable. I want to thank all of our speakers today for joining us and the growers who are online, and also Allison for putting the program together.
Thank you, Tom. Yeah, I think without further ado, we can pass things off to, I guess I already kind of gave the agenda in advance, so we'll pass things off to Jyostna to get us started talking about flooding. I'll stop sharing my screen so you can begin.
Thank you, Alison. Let me see.
And I just again want to thank you for hopping in on almost, a day and a half notice. So thank you for being able to give us good information. Just ready, turn on the faucet, and here's our good info.
Yeah, thank you. Hello, everyone. I'm Jyostna Mura. So Amaya is on travel. That's why I kind of decided to give this talk. So today I'm going to talk about my research on winter and spring floods, effects on dissolved oxygen and carbohydrates in cranberry. This research is funded by Wisconsin Cranberry Board. So I'm still doing it's not complete research. So I have only some preliminary data that I want to share with you all. Thank you. So let's talk about what happens under flood conditions. There are several consequences. I'm going to talk about what happens at the soil level and at the plant level. As you all know, when you flood, the soil gets compact and it changes the pH of the soil and also its electron exchange capacity. Overall, it causes oxygen deficiency in the soil. So at the plant level, we can see obviously plants are under water. So they reduce their photosynthesis and there won't be any root permeability and no transpiration, so no nutrient uptake. And so because there will be an oxygen deficiency, plants move from aerobic respiration to anaerobic respiration. That causes some metabolic changes in the plant. And some crop species, they undergo anatomical changes as an adaptation mechanism. So some of these changes are some of the anatomical changes, as you can see here, some plants, they develop lenticels on the stems. So that will allow them to have oxygen. And some cells, some plants, they develop parenchyma in their leaves, stems, and roots. So they develop large spaces in between the cells so that they can hold so much oxygen. And some crop species, they develop adventitious roots. We don't know about the cranberry because nobody has studied. Some crop species, they develop lateral branching as a mechanism so that they can reach out of the flood and then get the oxygen. So another mechanism, this is adopted by almost all crop species. And for all abiotic stresses, not only for the flood, even they are under the cold stress or flood stress or any stress, in general, they produce ethylene hormone. So this ethylene hormone under small quantities, it will help the plant, to withstand the stress and to develop some adaptation mechanisms. But if they produce larger quantities, we don't know, in some crops, it causes yellowing of the leaves and leaf drop. So for cranberry, we don't know how much is detrimental, how much causes leaf drop, how much is good, how much is bad. So that's what we are trying to investigate with Wisconsin Cranberry Board funding. So overall, this is what we are doing in our lab. So when cranberries are underwater, and also like, as you can see, like there are so many microbes in the soil and algae and other plants, they all uses some oxygen. So over the period, it causes oxygen depletion in the water. And from the previous studies, we know pretty much like 40% of the oxygen are 4 milligrams per liter of the oxygen in the flood causes damage to cranberry plants. So in general, like plants move from aerobic respiration to anaerobic respiration. Aerobic respiration means they uses water and they uses carbohydrate and they produce energy, good enough energy. But when they move to the anaerobic respiration where there is no oxygen, plant uses like more carbohydrates to produce like some energy so that they can survive. So even though they use like a lot of carbohydrates, they were able to produce only less energy. And the byproduct of this is ethanol, which is not good for the plant, actually. And overall, like using these carbohydrates causes the plant to have low carbohydrates. So because these carbohydrates are required for the growing season, to grow buds and plant growth. So as I said, like as an adaptive mechanism, plant produces ethylene. So, and if this ethylene is in low quantities, it helps with the stress acclimation. But if the ethylene is produced in high levels, that may result in leaf drop. So we do know like some leaf drop in cranberries after we remove the flood. But I don't know if that is because ethylene is also produced due to cold stress and winter damage also. So we don't know if this is because of flood or because of cold. That's what we are trying to find out. So overall, like what causes low oxygen levels in the plant in the water? So I got this figure from NOAA, National Oceanic and Atmospheric Administration, where they say like there are two parameters that affects the dissolved oxygen levels in the water. One is temperature. As you can see, as the temperature goes high, warmer temperature causes low dissolved oxygen. And water quality also plays role for dissolved oxygen levels in the water. So I know like not all of the growers uses dissolved oxygen meters, but we still did some research if they want to use them. So we purchased like different price ranges of dissolved oxygen meter to see if they read the same. So most of them read more or less same, irrespective of the price. The price differences are mostly for the sensors. If you want to measure for the spring flood, all of them work really well. But for the winter flood, only some of them work, like 700 to 900 and 1841. They are working well. Rest of them, like they either freeze or break down actually. So overall message is like any dissolved oxygen meter works for spring floods very well. So not for the winter floods. So, and also we measured like what happens to the dissolved oxygen level when you form the ice. So as you can see, day one, we measured the dissolved oxygen levels of the water and we found like 13 milligrams per liter, which is a good amount, but which is not critical. Like for cranberry, it is 4 mg per liter that is critical. But as the ice formed, we measured again on day 8 by making a hole underneath the ice. So you can see like the dissolved oxygen levels with the leftover water underneath the ice, it really went down, like decreased. So not only one marsh, we noticed that in several marshes, like, but we still have to work on like how much That is because I believe like it's not because of the flood, it's because of the ice formation. We have to see like how much thickness of the ice is going to affect the dissolved oxygen level under the ice layer. So, but overall, we found like in all of the marshes that we measured, after day 8, DO level goes down during winter floods. So, and we measured dissolved oxygen levels during spring floods also. So for spring floods, we found like on an average we measured in 4 marshes, and most of the average is like 11 mg per liter, which is good for Cranberry. So, but this is basically temperature dependent and also water source dependent. So basically, like if the temperatures are low, it is good. You will have like good oxygen levels. That's not going to damage our cranberry plants. But we also noticed like once you remove the flood, you will find like small puddles, like here in the picture you can see. So we found those puddle reads like very low. That means like if you leave them for longer time, they might damage the root, and also it causes like yellowing of the leaf and leaf drop. So when after flood removal, we check like most of the beds, but beds are not consistent. Some places you will find, dry spots, and some places you will find these small puddles. So just want to let you know, like these reads like very low DO level that can damage the root and also might cause leaf drop. So, and this one is from the previous studies by Sean Stephan. So as you can see, like with his study, I think like he measured in 35 marshes how the spring flood affects the DO level. What he found was there was no like, as I said, like spring floods have like a good amount of oxygen levels. You can see that from this picture, from this figure. But as the temperature goes up, you'll find like the DO levels goes down. So we have to watch for the warmer temperature and how that is affecting the DO levels. So overall, his message was like Wisconsin reservoirs are well oxygenated. So spring floods are okay. They have enough oxygen, but need to check for the temperature. If the temperatures are warmer, like we may have to remove the flood and re-flood them. So, and also like we measured, these are readings from 2023 winter. As you can see, like these are readings under ice after 7 days. After 7 days of ice formation, you can see like some of the marshes have really low DO levels under the ice, and some of them have like very high levels of oxygen. There is a variation across marshes, but we don't know what is this causing. It might be water quality or ice thickness formation. We are still investigating this. But when it comes to these readings are from the March, after ice melted, like March 1st week, as you can see, like all of the marshes have like good amount of DO levels. So if the temperatures are fine, so I believe like it's not going to affect any dissolved oxygen in the water. And these are the carbohydrates that we measure during winter and spring temperatures during flooding. This is kind of a greenhouse experiment. Like as you can see, like if the water temperature is 39, which is low temperature, so it's not causing any carbohydrate depletion. It caused the carbohydrate depletion, but not as much as 68F. So, and also for the starch, you can see like 68F had more carbohydrate depletion than 39 temperatures. So overall, what we found is if the temperature of water temperature is warmer, that causes depletion in the carbohydrates faster. So we didn't measure plant oxygen levels here, but we believe like this is because of lower oxygen levels in the plant. So we are still investigating the plant oxygen levels, but cold temperatures are not causing any damage, like are not causing depletion in the plant carbohydrate levels that much as warm temperatures. And also we measured ethylene during winter floods. We still have to measure spring flood ethylene levels. As you can see, like ethylene levels were increased. I think like day 4, the plant samples we collected have a lot of ethylene. But we found like after ice off, like we found as much as ethylene as day 2. Overall, like what we found that flood has an effect, flood and water, cold temperature, we believe like has an effect on ethylene levels. But we don't know like how much amount of the ethylene in the plant causes leaf drop. We are still working on it. So this is from a previous study again. As you can see, like if the spring flood has an effect on plant parameters, yes, if the temperatures are warmer and if the flood is prolonged flooding, then you can notice effect on the plant parameters like chlorophyll and hooks and flowers per upright. They all drop if the flood is prolonged. So some cultivars responded to the temperature and some cultivars didn't. As you can see, Stevens' upright length was not affected, but however, GH1 and Benlier had an effect on the upright length. So this study tells like, plant response depends on like floodwater temperature and duration of the flood. So conclusion from these studies are like winter flood causes dissolved oxygen depletion after ice formation. Spring oxygen depletion depends on temperatures. If the temperatures are warmer, that causes like low dissolved oxygen levels. Flooded plants produce acetylene, but we are still investigating how much causes leaf drop. And also we are investigating like warm flood water temperature, how they deplete the carbohydrates. Thank you. Any questions?
I think actually we've got a couple of questions that came in, and I've got a couple of minutes here for questions directly for you. Jocelyn asks, what was the ice thickness? And I think that was in the experiment that you were showing, I think from Sean's, that had at certain length of flood. I don't know if you have that answer offhand.
No, I don't have ice thickness. Actually, that was the same question I was asked in the board meeting also. We are, I think like that ice thickness has an effect on winter flood dissolved oxygen levels. So next year we are going to investigate that.
Okay, so unfortunately that was research conducted in the past, but before Jyostna was here, and so that number didn't get recorded. So we'll get some future extra information. I had one question get sent in. Is there a difference in holding a surface flood in the spring versus having the vines totally submerge? The situation would be waiting for more frost to leave the soil.
We don't see, actually, we measured at the different levels. As I said, like if you leave puddles, then it will cause dissolved oxygen, low dissolved oxygen, and that can affect the roots. But we don't see much differences, submerged or merged actually. So small puddles are the ones that are affecting.
Okay, so surface versus submerged can both be good. Avoid leaving small puddles. I also had one question that was, if your cranberry soil has diatoms in it, will they introduce oxygen to the flooded bed to allow for a high dissolved oxygen level?
That's a good question.
Right?
I don't know that. I think they might use themselves oxygen.
Oh.
So
okay.
I have to check, like, I have no idea about it.
Yeah, but there's a possibility they'd even be consuming it. So that is a
They
good
might consume actually, yeah.
Yeah. All right, so Cass will follow up with you, have you run a couple of test tube experiments on that, I guess. We could probably easily grab soils and just do a benchtop experiment. Again, we're super grateful that you're able to jump on short notice and give us some good information. And as you see, when I start going into my questions, we'll have a few pieces of information there that those will line up and be pretty nicely dovetailed.